of ribozymes and their role in the origin of life. The Miller experiment was performed in the 1950s. He placed molecules that were believed to be a part of the Earth's early atmosphere in a small closed system. An electrical current was run through the system. After a week, 10 to 15% of the carbon had been turned into organic molecules, such as amino acids. More amino acids were found by Juan Oro in 1961. He also discovered the presence of adenine. Similar experiments found other DNA and RNA bases, which fueled more experiments. This brings up the questions: what other organic molecules played a role in the early development of life on Earth? How do we find these molecules? What could be the implication of the discovery of these molecules? And can these molecules be used in modern times?
A ribozyme, short for ribonucleic acid enzyme, is an RNA molecule which can act as a catalyst, as an enzyme does. They often use metal ions such as magnesium 2+ as cofactors. The term ribozyme was first used in a published paper in 1982 by Kelly Kruger. The diagram here shows some ribozyme structures. They possess either a hairpin or a hammerhead-shaped active center, as well as a second structure which allows it to cleave other RNA molecules at specific locations or sequences. It is now possible to create ribozymes which will purposefully cleave any RNA molecule. Ribozymes were discovered around the same time by two different men doing work in two different parts of the nation. Thomas Cech from the University of Colorado at Boulder, who was doing research on the removal of introns, and Sidney Altman, a professor at Yale University, who was doing research on the processing tRNA that are inside cells. A few of the known or naturally occurring ribozymes are listed here. The concept of ribozymes help avoid this question of which came first: the enzymes to carry out the orders of the cell, or the nucleic acids which transport the information required to make the enzyme. The first needs the second, and the second needs the first. The revolutionary idea that the RNA in a cell can itself act as a catalyst avoids this issue altogether. Second, the hypothesis that RNA serves as the genetic material as well as the structural and catalytic molecule is known as the RNA world hypothesis. It was proposed by Walter Gilbert, who won a Nobel Prize in 1980 for his work in determining the sequence of nucleotides in nucleic acids. This hypothesis was in opposition to the then idea that those two functions were split into DNA's and proteins' respective jobs. Thirdly, if ribozymes were in fact the primary molecular machines used in first Precambrian life, that means that ribozymes today could be considered living fossils of a life based primarily on nucleic acids. Ribozymes can be made to specifically cut any RNA molecule, and these artificial ribozymes may have pharmaceutical benefits and uses. One example of this is ribozymes that have been manufactured to cleave to HIV RNA in several different sites in order to reduce the effectiveness of the HIV virus. Results from the experiment were that the ribozymes were able to disable the AIDS virus up to 96%.
Dr. Nikolai Zenkin, a professor at Newcastle University, explored the stepwise process of transcription and translation by way of in vitro experimentation via test tubes. He hypothesized that codon-anticodon interactions evolved from RNA helicase activity to melt RNA duplexes, and that protein synthesis is a side product of helicase activity. Helicase activity is a group of molecular motors that are the driving forces of RNA metabolism. This force is what helps RNA polymerase read double-stranded RNA at the 5' untranslated end, separating the two strands into a pair of single strands. Protein synthesis is divided into three phases: imitation, elongation, and termination, where a functional component of the ribozyme binds to the mRNA via polypeptide, transcripting until it reaches a stop codon, and then termination. The following is a list of steps in preparation for facilitated protein synthesis. Plasmids were obtained and transformed into E. coli cells. These E. coli cells were mixed with various chemicals and grown in an orbital shaker. Several steps of buffering and centrifugation purified protein components. The goal to purifying these components was to exclude additional explanations of transcription from other cellular components. Purification of DNA polymerase and RNA polymerase prepared the DNA and RNA templates transcription. The 5 ends of a DNA template were tagged for easy transcription observation. Transcription and initiation mixture was incubated and centrifuged and added incrementally for optimal observation. The resulting concentration concentration of ribosomes and transcriptional complexes scaled up accordingly. Dr. Zenkin and his research team made two distinct observations linking transcription to the primordial RNA world. The first observation was that the 16S subunit ribosome of the rRNA binds on opposite ends of the 23S subunit ribosome on the tRNA, resulting in transpeptidation. This sets the stage for synthesis initiation. This binding suggests that helicase activity of the ribosome is an ancient property, most likely dating back to the RNA world. These particular components are involved with initiating protein synthesis to stabilize the correct codon-anticodon in the pairing on the A site of the ribosome via hydrogen bonding. The second observation is that the RNA helicase required for RNA world replication provides a link between helicase activity and ribosome replication. The requirement for an RNA helicase is explained by the behavior of ribozymes acting as single-stranded units. Replication of these ribozymes is an inherent property and creates double-stranded helices, which are then melted to produce another pair of single-stranded ribozymes. This perpetuating process evolved into the replicating RNA template that we know today. The following figure shows in vitro characterization of ribozyme translocation, transcription, and translation. Dr. Zenkin's research established a strong relationship between transcription of RNA templates in ancient RNA world, but there are still questions left unanswered. How did the random non-sense sequencing machinery evolve into templated protein synthesis? Were there any selective forces? Evolution does not have foresight. What were the evolutionary advantages for the intermediate steps leading up to the templated protein synthesis? Perhaps towards some different function in the ancient primordial RNA world?
Researchers Tracy Lincoln and Gerald Joyce from the Scripps Research Institute have synthesized RNA enzymes that can replicate without any aid from proteins or cellular components in a process that proceeds infinitely. The RNA world model postulates that RNA came before DNA because RNA can function both as a gene and an enzyme. In vitro evolution takes one of the RNA enzymes that has already developed in the lab that can perform basic chemistry of replication and improve it till it can perform perpetual self-replication. From the enzymes placed in the test tubes, the ones most adept at joining together pieces of RNA were obtained. Cross-replication is a process that begins with two enzymes, each with two subunits. Each subunit functions as a catalyst. The first enzyme binds with the two subunits of the second enzyme, and vice versa. The two enzymes assemble each other and hence is called cross-replication. The continuation of the process requires only a small starting amount of the two enzymes and a steady supply of subunits. After the success of the initial experiment, another one was carried out using 12 different cross-replicating pairs of enzymes. Some mutants occurred, but some changes also occurred that were advantageous to the chemicals in replicating. The ones that developed the advantageous trait dominated in numbers compared to those replicating without the trait. This is very akin to Darwinian evolution.